Cancer is a growing problem, killing 7.6 million people globally each year, according to the CDC. Cancer occurs when cells start to proliferate, to divide without restraint, forming tumors which eventually spread to different parts of the body. However, the body has a genetic mechanism to fight cancer, p53. p53 is a tumor suppressor protein that stops the cell cycle in response to cellular damage. It is also a transcription factor, a protein that regulates the expression of other genes called its targets. My experiment seeks to understand the role that three newly identified target genes, SIDT2, FLDA3, and ABHD4, play in tumor suppression. The question that I sought to answer was, in mouse lung tumor cells, is the expression of p53 target genes sufficient to trigger the p53 cell cycle arrest function? My hypothesis is that the expression of p53 targets is able to stop the cell cycle. Cells expressing the target genes must be shown to stop proliferation to support the hypothesis. Cell cycle arrest is observed by comparing the amount of proliferation in the target gene samples to a positive and negative control. If there is a statistical difference between the target gene samples and the negative control, then we know that cell cycle arrest has occurred. After counting 5 trials, I found that the average proliferation in FLDA3 samples was statistically different from the negative control, partly supporting the hypothesis. The other 2 genes failed the test. This finding tells us that FLDA3 can act like a tumor suppressor to stop the cell cycle. Once a method is developed to increase the expression of tumor suppressor genes like FLDA3 only in tumor cells, then our knowledge of p53 and its target genes could be translated into a therapeutic cure for cancer within our own genes.